I'm Dr. Double B, and this is my intro to cancer genetics and molecular pathology. The cells in the body normally exist in balance. This balance is maintained by a complex signaling pathway that tells the cell when to proliferate. There are two main types of proteins that control these pathways. One is the oncogenes that tells the cell to multiply. The other is the tumor suppressors that tells it to stop. Tumor suppressors also perform other functions like repairing damage to DNA. Cancer is in large part caused by mutations in these genes for these proteins that disrupt normal cell proliferation, leading to a population of cells that proliferates without control. This concept of cancer being caused by an accumulation of mutations is referred to as the multi-hit hypothesis, demonstrated in this video. Wait, no, sorry, this is the wrong multi-hit hypothesis. This is Chuck Norris's multi-hit hypothesis. I have the right video here somewhere. Yes. An early change in colorectal cancers is loss of the tumor suppressor APC. At this point, the region of colon may have low-grade dysplasia and form an early adenoma. Mutations in KRAS and oncogene can lead to progression. The low-grade dysplasia can progress to high-grade dysplasia, and loss of P53 is a step in the progression to adenocarcinoma. In concept, cancer progresses from low-grade dysplasia to high-grade dysplasia to a well-differentiated cancer to a poorly differentiated cancer with a potential for metastasis. And all of these changes are driven by mutations in these cancer-related genes. Mutations can be caused by errors made by DNA polymerase at a rate of about 1 in 100 million per base per cell generation, or they can also be caused by sources of DNA damage like UV light, radiation, or carcinogens.
I need plenty of wholesome, nutritious alcohol. The chemical energy keeps my fuel cells charged.
What are the cigars for?
They make me look cool.
As an example, the protein P53 is the tumor suppressor most mutated in cancers. It is often referred to as the guardian of the genome, since it has many important roles in preventing cancer. First, it can activate DNA repair proteins in response to DNA damage. Second, it can arrest cell growth in an effort to allow the cell to repair any damage before dividing. Third, if all else fails, it can initiate apoptosis, in which the cell destroys itself, nobly preventing it from potentially leading to cancer. In many cancers, the gene for P53 is defective and cannot perform these functions. This could be due to a point mutation, which changes an amino acid to another, potentially damaging the protein. It could also be due to a nonsense mutation, which terminates the protein early by creating a stop codon. A deletion can remove an amino acid, or an insertion can add them. Insertions and deletions, also known as indels, can also cause a frame shift, which changes all subsequent amino acids. The gene could also be deleted, or the protein not expressed. We can look at p53 expression by immunohistochemistry and may see increased expression of a non-functioning mutant protein or complete loss. One of the major cell proliferation pathways is the EGFR pathway, which stands for the epidermal growth factor receptor. EGFR is a tyrosine kinase receptor which activates downstream cell proliferation pathways. This normally occurs after binding of a growth factor activates the receptor. The downstream oncogenes include KRAS, which stands for Kirsten rat sarcoma viral oncogene homolog. Sorry, a lot of these have useless names, and others like BRAF, mTOR, and AKT. Activation of this pathway encourages a cell to multiply, which could normally occur in response to injury or just as part of normal cell turnover. But certain mutations can cause these proteins to be active without any normal signals. One famous oncogene mutation is the BRAF V600E, seen in melanoma and many other cancers. The numbering means that it's a mutation of the 600th amino acid, and it changes the normal valine residue to a glutamate. This corresponds to a nucleotide change of T to A at position 1799, if you're keeping track. It's an activating mutation, meaning it causes the protein to be turned on regardless of the normal EGFR pathway. As a general rule, there aren't many mutations that will do this, make a protein more active than usual. Here's a graphic of the mutation distribution of BRAF in cancers, per COSMIC, the catalog of somatic mutations in cancer. As you can see, it's mostly just this one mutation along with a few rarer ones because there aren't many ways to overactivate a protein. The opposite goes for tumor suppressors. Look at p53 in comparison. Many mutations, insertions, deletions, and copy number changes can damage or eliminate the protein and cause cancer. When the BRAF V600E mutation is present in melanomas, it can be targeted with specific drugs, such as vemurafenib, which stands for V600E mutated BRAF inhibitor. The drug inhibits the mutated BRAF by targeting the ATP binding site, slowing tumor progression. And as a bit of trivia, treatment with this drug causes keratoacanthomas, a type of skin tumor, presumably by disrupting the normal cell signaling pathway. Now, the tricky part is BRAF mutations aren't just seen in melanomas or even just malignant cancers. They're also seen in benign nevi, for instance, so it must be in combination with other changes, genetic or otherwise, that it causes melanoma. Here's a list of some other tumors and syndromes BRAF mutations are seen in. Now, as you can see, it's still going. Okay, all right, as you can see, the same genetic change can be seen in different cancer types, and the type would depend on the cell type in which the mutation arose, as well as other factors. Let's talk a little more about melanoma. Different mutations are also seen, and interestingly, they depend on the site in which the melanoma arose. Melanomas on acral surfaces tend to have more NRAS and KIT mutations, and uveal melanomas have GNA11 and GNAQ mutations, part of the G protein pathway. Colorectal cancers have a number of important mutations. High-stage cancers can be treated with cetuximab, slowing cell proliferation by shutting down this pathway. But think about our last case of the BRAF V600E. If the pathway is already overactive beyond the point of the receptor, then the drug isn't going to be effective. Activating mutations in KRAS, NRAS, and probably BRAF indicate that such antibodies shouldn't be used. The other major molecular change in colorectal cancers is referred to as microsatellite instability or mismatch repair protein deficiency. The mismatch repair, or MMR proteins, are a set of tumor suppressors that predictably repair mismatches in DNA. The ones we regularly test for are MLH1, PMS2, MSH2, and MSH6. If there's a loss of expression of any of these proteins, a cancer can result. We can detect expression of these 4 proteins by immunohistochemistry, and if there's a loss of the protein, we'll see loss of expression in the tumor, but not the normal tissue. PMS2 depends on MLH1 to function, so if there's a loss of MLH1, we'll also see a loss of PMS2. Likewise, MSH6 depends on MSH2, so the same goes for them. Microsatellite instability, or MSI, is the result of a loss of these MMR proteins. Microsatellites are regions of DNA where the same 1 or 2 nucleotides repeat. Because they can shift and still be complementary, the DNA polymerase can slip while copying them. Normally, this mistake would be fixed by the MMR proteins, but if there's an MMR deficiency, the mistake will remain, resulting in a change in the number of repeats in the daughter cells. We can perform PCR around these regions and see if the tumor has different microsatellite length than the normal tissue. We normally test 5 of these microsatellites, and if 2 or more are different, we call it MSI high, or microsatellite unstable. Now, the traditional importance of testing for these is in looking for Lynch syndrome, or hereditary non-polyposis colorectal cancer syndrome, or HNPCC. This syndrome is caused by mutations in the MMR proteins carried through families that significantly increase a person's risk for cancer, mainly colorectal, but also endometrial and others. MSI high, or MMR loss, may suggest Lynch syndrome, though not always. Many cases with MLH1 loss are actually due to spontaneous methylation of the MLH1 promoter, which turns off expression rather than being caused by a germline mutation. You can test for this promoter methylation, or you can actually test for BRAF mutations, as 50% of these cases will also have a BRAF V600E, though if it's negative, it doesn't really help. Now, breast cancer has a few classic molecular changes that are important to treatment. The breast duct cells respond to estrogen and progesterone, which help drive cell proliferation. The cancer can potentially be treated with estrogen antagonists, slowing cell growth. First, we test the cells to make sure they're actually expressing the estrogen receptor and progesterone receptor. One group of breast cancers is driven by amplification of an oncogene, the human epidermal growth factor receptor 2, or HER2. The gene itself, technically called ERB2, is amplified, leading to increased production of the HER2 protein on the surface of the cell. This in turn leads to more downstream activation of oncogenes and cell proliferation, leading to a breast cancer. One way to detect this is with immunohistochemistry, which, like all IHC, is simply an antibody to the protein that can be detected with a dye. We then score these results on the staining scale of 0 to 3+, with 3+ being positive. The other way to look for HER2 amplification is to look for the gene directly through fluorescence in situ hybridization, or FISH. FISH uses a fluorescent DNA probe complementary to the target, in this case HER2. In a normal cell, you'd only see 2 copies of HER2 per cell, but in a case of amplified HER2, you would see many more copies, potentially as high as 30 or more. The value of finding this amplification is that the patient can be treated with an antibody to HER2, such as trastuzumab, aka Herceptin, which will shut down this pathway and slow the tumor. Leukemias and lymphomas are known for being driven by translocations between chromosomes, or within them. As one example, Burkitt's lymphoma is driven by fusions of the oncogene c-Myc and another gene, either IgH, Ig-kappa, or Ig-lambda. The latter genes are immunoglobulin genes that are expressed in very large amounts in B lymphocytes to produce antibodies. If a translocation brings together an immunoglobulin promoter with an oncogene, the oncogene can be overexpressed, causing a cancer. A translocation can also create a fusion protein, a chimera, that includes two separate proteins together. If one of them is an oncogene, it may become overactive, such as the BCR-ABL fusion in chronic myelogenous leukemia. This fusion can be treated with imatinib, aka Gleevec, a tyrosine kinase inhibitor that targets the aberrant protein. Translocations can be detected by FISH, for example, by a break-apart probe, when two signals that are normally adjacent are broken apart by this translocation. A more complex version, the dual-color dual fusion probe, has different color probes that span both breakpoints, so when a fusion occurs, these two colors are brought together. The karyotype is another broader technique that can look for any large chromosomal change. This requires live growing cells. They're given a mitogen to promote growth. Mitosis is then arrested in metaphase. The cells are lysed and the chromosomes are stained and viewed under a microscope. From here, the individual chromosomes are basically photoshopped to give us the nice lineup we're used to seeing. As an example, here's a karyotype of the 922 translocation in CML, where you can visualize the exchange of the pieces of the chromosomes. And here's an example of 814 in Burkitt's. Lung cancers are driven by a number of treatable changes. The most common are alterations in EGFR, including mutations, insertions, and deletions. These cause it to be constitutively active, constantly promoting cell proliferation. If this is the case, EGFR can be targeted with tyrosine kinase inhibitors, molecules which inhibit its activity. Here's a video of a patient I once saw with such a mutation.
Thank you for coming in again, Mr. White.
Yeah.
I've reviewed your biopsies, and you have a stage 4 adenocarcinoma, which is a tumor more common in non-smokers.
Oh my god.
Yes, it's very serious, but I've sequenced the EGFR gene in your tumor, and you have a mutation in the tyrosine kinase domain.
Wow.
Well, what this means is that we can treat it with targeted drugs. Now, unfortunately, these drugs cost upwards of $100,000.
Jesus.
Now, Walt, I know that you've been cooking meth to pay for your treatments.
Wait a minute, Walt, when did this happen? Why didn't you tell me?
Now, everyone calm down. I have a much better solution. Maybe you weren't paying attention. What I said was, your drugs are going to cost upwards of $100,000. Forget meth. Partner with me and cook chemo.
Mutations in BRAF are also seen and may have specific therapies. Fusions in ALK and ROS, both tyrosine kinase receptors, can drive cancer and both have specific therapies. The fusions can be detected by FISH or by immunohistochemistry, looking for overexpression. HER2 overexpression or mutations are rarely seen, amplification of the MET oncogene, RET fusions, and more, but one deserves special attention, and that is PD-L1. One of the roles of the immune system is to wipe out cancer and cells that may lead to cancer. Some tumor cells avoid this by expressing PD-L1 or programmed death ligand 1, which binds to PD-1 or programmed cell death protein 1 on immune cells. This interaction can shut them down. Let me explain.
Okay, and how old are you, McLovin?
Old enough.
Old enough for what?
To party.
Can I see your ID?
Yeah, I think I have it.
Here, we have a tumor cell faced with an immune response. The tumor cell can present the PD-L1 or its fake ID to the immune cells, making them think all is well.
You're an organ donor.
What?
I didn't want to be one, but my wife insisted. I always give him shit for it, too. I say it all the time.
A relatively new set of drugs called immune checkpoint inhibitors bind PD-1 to prevent the shutdown of the immune cells. Before using these drugs, in some tumor types such as lung, it's useful to know if the tumor cells actually express PD-L1. This can be done by immunohistochemistry, and there are complicated rules on judging the results that depend on the tumor site. On the other hand, use of these inhibitors in some sites, such as melanoma, doesn't require this testing at all. But there's more. Part of the way an immune cell might recognize a cancer is if it expresses new antigens that don't exist on normal cells. Mutations can cause these new antigens, so you might expect a cancer with more mutations to have a better response when immune checkpoint inhibitors are used. And you might expect a tumor with a mismatch repair deficiency to have more mutations, and you'd be right. An immune checkpoint inhibitor, Keytruda, is approved for any solid tumor with mismatch repair deficiency or microsatellite instability. Same idea. Tumors with increased mutational burden, or TMB, also have better response to these drugs. The last thing I want to talk about today is inherited cancer syndromes. A classic example is familial adenomatous polyposis, or FAP. Be careful Googling that nowadays. Remember the steps leading to colorectal cancer? The first was a damaging mutation in APC, or adenomatous polyposis coli, named after this syndrome. It's a tumor suppressor that, among other things, inactivates the beta-catenin oncogene. Some people carry an inactive APC gene from birth. This jumpstarts the pathway, leading to thousands of polyps and inevitably leading to cancer. Heritable syndromes are classified as either dominant or submissive. Wait, no, that's not right. It's recessive. Sorry, I must have been thinking of something else. Dominant syndromes only require one abnormal allele to cause a disease, whereas submissive or recessive syndromes require two abnormal alleles. If you look at all of the cancer syndromes, you'll find that almost all of them are caused by mutations in tumor suppressors, not oncogenes, and almost all have autosomal dominant inheritance. Why is that? Well, if you think about an activating mutation in an oncogene, it's immediately going to cause abnormal cell growth. This couldn't exist in every cell in the body, or it would be incompatible with life. On the other hand, one working copy of a tumor suppressor is generally enough to do its job, such as repairing DNA. However, over a lifetime, there's so much random damage to DNA that in some cells, the normal copy will inevitably be knocked out as well, and these cells can progress to cancer. This is called the two-hit hypothesis, and as you might guess, inherited mutations in both copies of a tumor suppressor are almost never seen. Retinoblastoma, a rare cancer of the retinal cells, is a classic example. This tumor occurs when a person has loss of both copies of retinoblastoma, or RB, a tumor suppressor protein. A person without an inherited mutation has a very low risk of retinoblastoma, about 0.004%, because they would have to have two damaging mutations, one after the other, in the same line of cells. But someone who carries a germline mutation in the RB gene only has to have one more knockout mutation, so they have a 90% risk. So, in a sense, cancer is a statistical phenomenon with mutations occurring randomly over a lifetime, sometimes in the right combination to cause cancer. And this risk is exponentially greater for someone carrying a germline mutation in a tumor suppressor. That's all for today, and be sure to like and subscribe if you want to see more.